Hello and welcome to Just the Facts podcast. This is your April 10th, 2020 update. I'm your host, Dr. Michael Bechill, and here we're to tell you about all things coronavirus, what's going on around the world, what's happening in your neighborhood, but most of all, just the facts. Today we have an outstanding episode that was actually created in its entirety by my virology class at the University of St. Francis. And today's podcast actually turned out to be so long that I'm splitting it into 3 episodes. So over these next 3 episodes, we're going to cover everything from updated numbers, local information, general vaccine information, SARS-CoV-2 research, updates on policies, and answering your questions. But to start off with this first segment, today what we're going to be answering is, what is the coronavirus vaccine? We keep hearing about it in the news. It's being developed. What is a vaccine? How does it work? And most importantly, when will we be getting ours? So this is what we're going to cover in this episode. You can look forward to the other content being covered in the episodes in the next 2 days. A 3-for-1 deal. I think it's a win-win situation for everyone. So here we go, and let's talk a little bit about vaccines. I'm very privileged to welcome to the podcast 3 members of my virology class at the University of St. Francis. So you're going to get to hear from all 3 of them today. From left to right on your screen right now, you can see Emma Applegate, Gerka, and Sam Rhoades. And they're going to be talking to us specifically about the SARS-CoV-2 vaccine and answering that burning question that's in the media now: when will it arrive? So first up to talk to us today is Emma Applegate. She is a senior and joining us today on the phone from South Bend, Indiana. She is finishing up her pre-PA degree right now and will be starting PA school here in just a matter of weeks. Welcome to the program, Emma.
Thank you very much, Dr. Bechill. I appreciate the introduction. So I'm going to begin by kind of going over some general vaccine information. The very first thing I'm going to touch on is, why are vaccines so important? So vaccines are important because they help break the chain of transmission. They allow immunity in the absence of a virus. They increase human longevity, which is seen in the top picture. You can see that the age that humans are living to has increased substantially over the years, mainly due to medical advancements. And they can also create immune memory.
Excellent. So I did notice on the right-hand side of this slide here that it mentions this herd immunity. Could you please elaborate on what this herd immunity is and why it's so important?
Yeah, of course, and excellent question. So herd immunity is the concept by which vaccinating a certain amount of the population will decrease the spread of the virus, due to the fact that the virus will not be able to infect as many people in the community. The number of people that need to be vaccinated for herd immunity to work depends on the virus and the population. An interesting side note on this concept was mentioned by Chen from the University of Toledo Medical Center. So some countries, such as Brazil, Sweden, and some African countries, are trying this concept of herd immunity against the coronavirus, rather than the stay-at-home order like the United States is implementing. It is highly controversial because it requires a large amount of the population to get infected. This way allows for the majority of the population to get infected, recover, and produce antibodies of their own against the virus, with the idea that they will not be able to get sick again due to the circulating antibodies created. If they can't get sick again, they cannot infect others later on. The idea of this is to have the disease peak earlier, but also end shorter, which would lessen the social and economic impact. An important aspect for the herd immunity is the basic reproduction number. Coronavirus has a basic reproduction number of around 2 to 3. So for every 1 person that gets sick, they'll infect approximately 2 to 3 people. So it would require about 60 to 70% of the population to get infected and have circulating antibodies. The only people that would remain in this quarantine would be the susceptible populations, which is the elderly and the immunocompromised. Chen points out that there's no evidence as of right now to know if this would truly work, and if it would fail, it would cause a lot more death. Another thing to keep in mind with this concept is that research shows that this virus mutates at a very high rate. So there's no evidence that if a person gets infected with the coronavirus, survives, and recovers, they will also be immune to another strain. Chen, my source for this information, does not think it's ethical to put a price on human life and mentions that you really cannot have a completely functioning society with dead citizens. He does make the important comment that although this may not be the solution to the current pandemic, it is important when a vaccine is developed to figure out how many people must receive the vaccine for herd immunity to be established. So this kind of leads me into the next topic, which is what do vaccines do? And vaccines, similar to herd immunity, they allow for circulating antibodies to be produced against the virus, but rather unlike herd immunity, this is in the absence of the virus. So they're able to expose the body's immune system to viral proteins to elicit a response and create memory. The picture shows that it typically takes about 2 weeks to begin producing antibodies. And on the right side of the picture shows that the immunological memory created can last for years.
Excellent.
So next, the adaptive immune response can take up to 2 weeks to respond, but a vaccine increases this response time. So the picture on the left can show the importance of the vaccine and how it works. The top picture shows the vaccine being entered into the body at a weakened state, the viruses, and the second picture shows that the body makes antibodies to fight the invaders. These antibodies can create immunological memory that won't go away for many, many years. Therefore, in the third picture, if the actual disease ever enters the body in a natural way, the body will have already had antibodies or fighters to fight the infection and clear it quicker, typically before symptoms can even occur.
Excellent. So, you've explained this first vaccine and how it was developed, but for obviously reasons, we can't, really go on with this infection process. So how were vaccines really started off? How did we get into delivering these first vaccines?
Yeah, okay. So the first vaccine, it was created by Edward Jenner in 1796, and it was a vaccine against smallpox. And he noticed that milkmaids who got cowpox would not get the highly fatal smallpox disease. So he took viral particles from a milkmaid and actually inoculated a very young boy with it. The boy got fever-like symptoms, which went away within a couple of days. And 2 weeks after his symptoms subsided, he inoculated the boy with smallpox. And obviously, we cannot do this anymore, but the boy had antibodies to cowpox and therefore did not become sick from smallpox. Due to this was the very first vaccine that was created, and smallpox, due to mass vaccination efforts, was officially eradicated in 1979. Now, there are some really important characteristics of viruses, which is what I'm going to go into next. And some of these include that it must elicit an immune response. The person who receives it needs to be able to be protected in the future, otherwise it does no good. It cannot incite disease, and some other small things that need to be taken into consideration are storage. Most vaccines have to be stored at very cold temperatures. There has to be minimal side effects. There has to be easy delivery and very low cost because they have to be mass produced. So there's two types of vaccines. There's the live attenuated vaccine, and this vaccine virus is typically capable of growing, inducing immunity, but is incapable of causing disease. And the other type of vaccine is inactivated, and this type of vaccine cannot cause disease because it's killed, the genome of it is killed by either heat or chemical. So the first one I'm going to go into a little bit more detail is the live attenuated vaccine. So this is typically weakened through repeated culturing, and you typically have immunity after one dose. The virus is typically modified in a lab, so it can't cause disease. And there is a problem with it, and that is that circulating antibodies in the blood can often cause the vaccine to be ineffective. Some examples of live vaccines are yellow fever, influenza, the nasal flu mist, and MMR.
Excellent. Now, you did mention that live attenuated vaccines can have some serious complications. What are these complications that you see with the live vaccines, and who's particularly susceptible to them?
Yeah, of course. So in cases of immunodeficiency, such as HIV and leukemia, live vaccines can cause severe complications such as pneumonia, respiratory failure, and can exacerbate other diseases that are underlying.
Interesting. So it's kind of the
So
trade-off if you get that robust immune response. The trade-off is you might have some of these complications.
correct, yep.
So not everybody can get these live vaccines. So I'm assuming there's the alternative.
Yes, and the alternative is the inactivated vaccine. And the inactivated vaccine cannot cause disease like the activated, even in immunodeficient people, because it cannot replicate. Because as you can see in the lower picture, the genome is destroyed and therefore incapable of replication. So inactivated vaccines can be made from whole viruses or parts of viruses, and they oftentimes require multiple doses. Another interesting thing about inactivated vaccines is they're not affected by circulating antibodies in the blood. So another problem with the inactivated vaccine is that antibody titers typically decrease with time. Some common examples of inactivated vaccines are rabies, the influenza flu shot, and diphtheria.
Now, I understand that there are some problems with it, needing the booster shots, so to speak. But if there are no side effects, then why do we even bother using the alternative, those live attenuated vaccines? Why do we even do anything with the attenuated vaccines if these inactive vaccines, don't cause illness?
Yeah, for sure. So we typically use these attenuated vaccines because they are more effective at exposing your immune system to the virus, which therefore makes the body's immune system produce more effective antibodies. And the majority of the population doesn't have an issue with attenuated vaccines. And as previously mentioned, and as you mentioned, attenuated vaccines typically only require one dose, which can reduce the cost of making the vaccine since you don't have to make it multiple times. Not to mention, many people today are not very compliant when it comes to vaccination schedules. So the likelihood of being able to deliver one vaccine is higher than the ability to deliver one at a time and hope the patient comes back for their booster shot.
Excellent.
So there's two types of immunity that both of these vaccines can offer. The first is active immunity, and active immunity allows for long-term protection. It's the most effective type of immunity because it's long-term, and it's typically due to a modified form of the pathogen. The second is passive immunity, which is temporary, and this is where you insert products of the immune response, such as antibodies, into a host. These antibodies are typically either cultured and modified in a lab, or they're made in the surrogate animal model and then inserted into humans. So the most common form of passive immunity is mother to fetus via placenta transfer.
Interesting. Well, thank you so much for that information about, vaccines in general. Why are we even getting them? Why they're important. I really appreciate that.
Yeah, no problem. Thank you for having me, Dr. Bechill.
No problem. And next on the show, I'm very happy to welcome Kara Gerka. So Kara Gerka is joining us from Fishers, Indiana. And she is also a graduating senior in my virology class and will also be starting PA school at the University of St. Francis here in a few weeks. Thank you so much for joining the program today, Kara.
Yeah, no problem. Thank you, Dr. Bechill, for that introduction. And so I'm just going to come in here and talk for a few slides, and then one of my classmates will come in and I'll sort of finish up at the end.
That sounds great. And so what are you going to start us off with today?
I'm first going to talk to you guys about some of the typical vaccine ingredients. So a lot of you are aware of the anti-vaccine movement. It's gained a lot of attention here in the media as of late. And if you're not, there's currently a percentage of the population that chooses not to get vaccinated. And they can choose not to get vaccinated for various reasons, but the most common reason is that they believe that typical vaccine ingredients cause harmful effects on the body. And this, of course, is not true. There have been numerous CDC-funded research studies that have proven that these vaccine ingredients are safe. And so what I'm going to do is just give you an example of each of these vaccine ingredients, tell you why they're needed so that if anyone ever comes up to you and makes a false claim, you can correct them and explain to them why each of these ingredients are necessary. And so the first typical vaccine ingredient is preservatives. So these may be things like thimerosal, which are added to prevent contamination of the vaccine. A vaccine may also contain adjuvants, and these are just chemicals such as aluminum salts that are added to increase the immune response of the vaccine and thus increase its effectiveness. Another ingredient that may increase vaccine effectiveness are sugars, otherwise known as stabilizers. Vaccine may also have residual cell culture materials. So these may be things like egg proteins that are present because some vaccines are made in chicken embryos. A vaccine may also have residual inactivating ingredients, so things like formaldehyde that are added to inactivate the virus while in production so that the virus doesn't cause disease when you're exposed to it in the vaccine. And then the typical vaccine may also include residual antibiotics. So these are things like neomycin, which are added to prevent bacterial contamination.
Excellent. So you got quite the ingredients list there, but I also heard you mention thimerosal and formaldehyde, and many anti-vaxxers or those that are vaccine adverse are also concerned about the negative effects of these ingredients. Could you maybe elaborate why we're even putting these ingredients into vaccines? Are they really necessary, and are they actually safe?
Yeah, absolutely. So I mentioned before that formaldehyde inactivates toxins while making a vaccine. And so formaldehyde is a compound made of carbon, oxygen, and hydrogen. So all elements that are naturally found in the body. And there have been some studies that in very, very high concentrations, they can cause deleterious effects. However, your body is going to be exposed to very, very low levels of formaldehyde in a vaccine. Your body produces much higher levels of formaldehyde naturally. And so we're really not going to see any negative effects as a result of formaldehyde exposure in vaccines. As for thimerosal, this is a mercury-based compound that is added to multi-dose vaccines to prevent contamination. And this has actually been removed from most vaccines except for the influenza vaccine. And so the main claim that started the anti-vaccine movement was that thimerosal exposure in vaccines led to autism. And since that claim was made, there have been 9 CDC-funded research studies that have investigated this link, and all found that thimerosal exposure does not lead to autism. So if you ever hear that, that is not true.
Yeah, there's no scientific evidence pointing to that at all. Thank you so much for that clarification. Now, now that we know that what's in the vaccine and kind of a better idea that, these ingredients are safe, they're necessary to get your immune response, could you tell me a little bit about what the normal vaccine development process looks like? And is this process suspected to be modified or different when we talk about specifically the SARS-CoV-2 vaccine?
Yeah, so that's a really good question. I think that's sort of the golden question right now. So the typical vaccine development process takes about 10 years from start to finish. The fastest vaccine that was ever produced was produced in 6 years. And this was the Ebola vaccine that was just approved actually in December of 2019. And the reason this vaccine was able to be approved so quickly was that the FDA granted researchers working on this vaccine breakthrough therapy designation and priority review. And so what breakthrough therapy designation is that for vaccines for severe or life-threatening diseases, the FDA provides more guidance for those researchers in vaccine development and helps to fast track those researchers through the developmental stages. And priority review, so once a vaccine has made it through clinical trials, the FDA reviews that vaccine application first. So normally there's a waiting time, and if the FDA grants that priority review, that is waived. And so for the case for SARS-CoV-2 vaccines, this process is expected to be expedited significantly. So instead of the vaccine development process taking 10 years for SARS-CoV-2, it's expected to take anywhere from 12 to 18 months. And so this is unprecedented, but what that means is that we're not going to have a vaccine to help aid us in this pandemic. And I'll talk a little bit more later about why it's still important that we produce a vaccine. So step one in this process is the exploratory stage, and this is the research intensive stage. So in this stage, scientists are trying to identify natural or synthetic antigens that can be used in the vaccine. And so just as a reminder, an antigen is that particle that preps your immune system so that your immune system is ready if it's ever exposed to the real disease-causing virus. And so if an antigen is selected, then the vaccine can enter into the preclinical stage, stage 2. And in this stage, the vaccine is tested on animals or cell culture. And scientists in this stage are trying to determine whether the vaccine causes any toxic effects. And if it's determined that the vaccine doesn't cause toxic effects, then the scientists can send an application into the FDA, which is the organization that's responsible for approving vaccines. And if that application is okay, then the researchers can begin stage 1 of clinical trials. And so in phase 1, small groups of people, anywhere between 40 and 100, receive the vaccine. And the researchers in this stage are trying to determine whether the vaccine's safe, determine correct dosage, route of admission, things like that. And this stage can last several months to a year. So all the vaccines that we're going to talk about today are in phase 1 of this clinical trials. So they still have a little while to go. If the vaccine passes this, then it can enter into phase 2 of clinical trials. And in this phase, hundreds of people receive the vaccine, including members of the more vulnerable populations. The goal is still the same in this stage. So researchers are still testing for safety, dosage, things like that. But this phase can take up to 2 years. So it's much more lengthy than the last one. If the vaccine passes this, then it can enter phase 3 of clinical trials. Once again, the goals are the same, but now thousands of people are receiving the vaccine. So you're really receiving a great wealth of information in this stage. And this stage can take anywhere from 2 to 3 years. And so if the vaccine passes through all phases of clinical trials, then it can enter into stage 3 or stage 4, and that's the regulatory review and approval. So the researchers send in this huge document that's 100,000 pages long to the FDA. And the FDA reviews it, makes sure that all the data gathered in each of the prior trials is okay. And if the FDA approves the drug then clinicians can prescribe that vaccine to patients and the drug can begin to get manufactured. And so even manufacturing takes time. So even if a SARS-CoV-2 vaccine is produced, it'll still take time for enough of that vaccine to be produced to vaccinate the whole world. So that's also important to take into consideration. And then even after everyone's received the vaccine, the FDA still does quality control and makes sure that the vaccine is still safe and updates the vaccine in case the virus that's in circulation is different from that which the vaccine confers protection against.
Excellent. Thank you so much for going over that process. And I know you'll be back here just in a couple minutes. And so thank you for kind of going over what the beginning of the process looks like.
Yeah, absolutely.
All right. Next, joining us on our program is Sam Rhoades. Sam Rhoades is joining us today from Fort Wayne, Indiana. He is a senior pre-PA major at the University of St. Francis. And if you're not noticing a theme yet, he also will be starting PA school here in a few weeks. And so thank you so much for joining us today on the program, Sam.
Thanks for that introduction, Dr. Bechill. It's awesome to be a part of something so cool that you have going on here.
I really appreciate it. And so today you're going to kind of talk to us a little bit more about vaccine development, and then maybe move into what we're looking at with the coronavirus vaccines, correct?
Most definitely. So the first slide I have here is kind of just to supplement a little bit of what Kara was talking about. So as she mentioned, it takes about an average of 10 years and $1 billion to create a vaccine. And as they're saying for the SARS-CoV-2 vaccines, that these could be created in as little as 18 months. And I think it's important for the viewers to understand why these vaccines could be created so quickly. And one of the main reasons that these vaccines have the prospect of being developed so quickly is due to our great technological advances in genomic sequencing. So shortly after the Chinese had this epidemic in their country, the SARS-CoV-2 genome was sequenced and made available to the public. And shortly after that, scientists in America could begin their work on these vaccines using genetic information alone. So what this means is that Emma talked about some inactivated vaccines earlier. And a couple of the vaccines that I'm going to talk about today are DNA and RNA subunit vaccines. And these vaccines are created solely from genetic information. And so, like I said, these vaccines are able to be produced so quickly because we don't have to try to culture the virus in cell culture, which can take many months or years to accomplish and really slow down the whole process. And also, I think it's important to note that many vaccines fail between phases 2 and 3 of clinical trials. And this is what people in the vaccine world call the valley of death. And none of our SARS-CoV-2 vaccines are even near this point yet.
Gotcha. So we're really right on the front end of this. So you talk about how the FDA is, putting these SARS-CoV-2 vaccines on the quote unquote fast track. Could you maybe briefly touch on this fast track and what it means for this production timeline for the vaccines that you're going to be talking about today?
Yeah, so just a little bit of update on the current vaccine information for our SARS-CoV-2. Like you said, the FDA has put this on the fast track. And this is an expedited process for drugs or biologics, such as vaccines, that are designed to treat serious or life-threatening conditions that address unmet medical needs. And so what this does is it allows for more frequent communication between the biotech company producing these vaccines and the FDA to get it approved quicker. And this is kind of what Kara was talking about earlier with Ebola and how that vaccine was able to be produced so quickly. And so what this does is, as she said, it makes it eligible for priority review. And this is where the FDA directs a majority of its resources towards the vaccine at hand in order to get it approved as quickly as possible. So you're looking at a timeline for approval after the vaccine goes through all of its stages of development of about 6 months versus what you'd expect 10 months for a standard review.
Excellent.
And another thing that's important to note is the fact of emergency use authorization. And this is something that the FDA commissioner has the power to grant. And he can allow unapproved medical products to be used in emergency to treat, diagnose, or prevent serious life-threatening conditions, such as the SARS-CoV-2 pandemic that we have going on right now. So if he deems that the perceived immediate medical benefits outweigh the possible unknown long-term side effects of these vaccines, he can make them immediately available for use in the public. And this has been done with many SARS-CoV-2 tests, a couple breakthrough medications, and other products such as ventilators. And so as you see on the top of this slide, we currently have 3 vaccines in phase 1 of clinical trials, and then there are about 59 others that are being developed.
Excellent. Now, are there risks associated if we were to use the emergency use authorization? Do you foresee any risks with that?
Most definitely. So like we said before, the vaccine approval process takes many years, an average about 10 years to produce these vaccines. And one of the reasons why it takes so long to produce a vaccine is because these scientists and these researchers are looking at all the possible dosages, all the possible side effects that could occur in these patient populations who they are giving the vaccines to. And so a great amount of research is being conducted when these vaccines are in these clinical trials for 10, 15 years to see the possible long-term side effects. They want to get a big picture idea of how these vaccines will affect people in the long term. So if we do produce a vaccine in as little as 12 or 18 months, like they're saying, it's really hard to understand and predict what possible side effects we could be seeing in these people.
Good. I think that's an important note. Now you have you have these 3 vaccines that are going into first-in-man trials. And I think you're going to talk to us about those, correct?
Yes, sir. Yep. So the first vaccine that we have here, this one has not entered phase 1 of clinical trials yet. They're in the process of applying for the investigational new drug approval, which is kind of like a precursor before this vaccine would enter phase 1 of clinical trials. And so like you see here, it is in development by the University of Pittsburgh School of Medicine. It is a subunit vaccine consisting of the SARS-CoV-2 spike protein. It is administered using microneedle technology.
Interesting. Now, I've seen many of these vaccines currently in production or development that are specifically going after this SARS-CoV-2 spike protein. Why are most or many of these vaccines being made to go against that protein? Why is everyone so focused on this spike protein?
Yeah, so I think that's a really good question. So if you look at the right side of your screen here, the kind of little cartoon drawing that we have, so the little particle on the left is a drawing of a rough drawing of the SARS-CoV-2 coronavirus. And on the right there, we have a picture of a human cell. And so if you could imagine those purple proteins, those purple spikes sticking off of the coronavirus, those are actually what we call the spike protein. And how this virus actually attaches and enters the human cell is that spike protein attaches to the ACE2 receptor on the surface of our human cell, and it fits exactly like a lock and key mechanism. And so once that protein connects to the receptor on the surface of our cell, it attaches, and then that allows the virus to enter and ultimately infect our cells. So if you can imagine somewhere in a lab at the University of Pittsburgh School of Medicine, these scientists are creating millions of these spike proteins, those purple proteins on the outside of that virus. And so what happens is once they inject these proteins into our body, our immune system can recognize this spike protein specifically, and we can mount a robust immune response against this protein and ultimately create antibodies against that spike protein. So if you can imagine a protein that we created that our body created against that spike protein on the outside, if you can imagine something bound to the apical part of that protein, then it would prevent it from attaching to the ACE2 receptor on our own cells, and ultimately that virus wouldn't be able to attach and then ultimately infect our cells, causing the disease that we see. And so I think an important thing to note about this vaccine that's being in development is that these researchers have seen these anti-SARS-CoV-2 spike proteins antibodies in our blood as early as 2 weeks post-vaccination. And so that is really promising for this vaccine.
Excellent. Now, looking at the bottom of this slide here, most vaccines are delivered by injection, but we kind of got this like gnarly looking patch that looks like it has a bunch of spikes sticking out of it. Could you maybe tell our viewers what they're trying to accomplish here at Pittsburgh with this type of delivery?
Yeah, so like I mentioned before, this vaccine is administered using microneedle technology. And so this is pretty cool, groundbreaking stuff. I believe they've tried it with a couple of influenza vaccines, but there's nothing available on the market as of right now that specifically uses this technology. And so like I said before, if you can imagine these scientists at the University of Pittsburgh School of Medicine, they're creating all these spike proteins. And what they do is they put a mold for those microneedles onto a centrifuge, which is a machine that spins stuff around really, really fast. And what happens is when they put that protein into that mold and spin it down really, really fast, that protein ultimately creates those spikes. And those spikes are on the underside of what appears to be a Band-Aid. And so what happens is the way that this vaccine is administered is you simply put this patch on your skin just like you would a Band-Aid, and those spike proteins are injected into your skin, which then allows your immune system to respond and mount a response. And they say that the microneedle technology is seemingly painless, and it only feels like a piece of Velcro is being rubbed up against your skin. And then after a couple of days, the needles dissolve into your skin, and you can never tell that anything was there.
Wow, that's very, very exciting. Awesome. So this we'll call this kind of like option one, this Pitcovax that we see coming out. But there are some alternatives. Can you tell us a little bit about this Moderna vaccine?
Yeah, so this actually is the first vaccine in the United States that entered phase 1 of clinical trials. Like you said, it is being developed by Moderna and the National Institute of Allergy and Infectious Disease. It entered phase 1 of clinical trials on March 16th at Kaiser Permanente Hospital in Seattle, Washington. And this is a RNA vaccine, so it's an inactivated vaccine, it's a subunit vaccine that consists of mRNA encoding that same SARS-CoV-2 spike protein.
Excellent. Now I notice kind of on this slide that it's an mRNA vaccine. What's the point of delivering mRNA? What is mRNA and why are we delivering it into these cells?
Yeah, so I think to understand how exactly this vaccine works, our viewers should understand what goes on in a typical cellular environment. So normally we have DNA in our nucleus of our cell, which makes up all of our genetic information. And when our cell wants to create a protein, this DNA undergoes a process called transcription, where the DNA is then converted into mRNA, which is also called messenger RNA. And this messenger RNA is kind of like a recipe, and it provides instructions for our cells on how to make certain proteins. And so while this protein may be created by the cell, displayed on the cell surface, or secreted by the cell, what also happens is that what we call this protein gets processed and presented on the surface of our cells. And so this is a normal cellular process because as these proteins are presented on the surface of our cells, it allows for cells of our immune system to kind of come around and survey these cells to see if they're producing any cancerous proteins, or in this case, any viral proteins that would indicate that our cells are infected. And so if you take a look at the right side of the picture here on the screen, if you can imagine a piece of mRNA coming in contact with our cell, being taken up by our cell, and then ultimately being released into the cytoplasm of our cell, which has all of our cellular machinery that are required to make proteins, this mRNA will actually guide our cell and instruct it to make the SARS-CoV-2 spike protein. And while this might kind of seem counterintuitive, like why are we producing a viral protein? The answer is that this protein will also be processed and presented on the surface of our cells. So then our immune cells can come around and they can survey these cells just as they normally would. And once they see this foreign antigen, this spike protein presented on the surface of our cells, then we can mount an immune response against that spike protein and create antibodies and then eliminate the infectivity of the virus, similar to how I demonstrated on the last slide.
Excellent. Very good. That's a very neat concept. It's one of the most modern ways of developing vaccines is using genetic information. Now, was a great example of an mRNA vaccine, but we also have some alternatives, don't we?
Yep. And that's what I'll kind of go over real quick on the next slide. So the next slide here, we have a vaccine that's called INO 4800. And this vaccine is actually the latest vaccine to enter clinical trials. It just entered phase 1 this past Monday, April 6th, and it is being tested out at the Perelman School of Medicine at the University of Pennsylvania and the Center for Pharmaceutical Research, which is in Kansas City, Missouri. And so, like you said, this is an alternative. This is the DNA vaccine, and it is administered using Selectra technology.
Interesting. Now, you had mentioned that this being a DNA vaccine, it's going to be different. So can you explain to our viewers how is a DNA vaccine different from the previous mRNA vaccine that we talked about on the last slide?
Yeah. So similar to the process that I explained in the last slide, about the whole creating proteins, processing, and then presenting them to cells of our immune system. This vaccine, we're pretty much just going to have an extra step, an extra intermediary step involved. And so if you take a look at the left side of the picture here, you can imagine that DNA is injected into our body. It comes in contact with the surface of our cell. It is then taken into our cell. But instead, this time, since it's DNA, it actually enters the nucleus of our cell. And then that's where we normally house all of our other genetic information. So this is just that extra intermediary step. The DNA that is initially injected into our bodies is then created or converted into the messenger mRNA, which then provides our cell with instructions on how to make the protein that is ultimately processed and presented on the surface of our cells to our immune system, and then we can mount an immune response against that. And like I previously said, this is administered using Selectra technology, which is a pretty cool technology. For the scientists that are out there watching, they probably know it also as what is called a gene gun. And so as this DNA is simultaneously being injected to our body, it literally looks like a little gun that delivers electrical pulses into our skin, into our body, while the DNA is being injected into our skin. And this helps create tiny pores in our cells so that our cells will actually take up this DNA better. And then it will just help with the overall efficacy of the vaccine and help our bodies respond to it much better.
Excellent. So we have several different kind of options out here. That's kind of exciting. Thank you so much, Sam, for showing us those three options as we move forward. I really appreciate you coming on the program today.
Thank you.
All right. And next, we have another kind of recombinant vaccine to talk about. And I think Kara is going to be rejoining us for this slide.
Yes, thank you. So the SARS-CoV-2 vaccine makes use of an adenovirus vector. And so this is the same platform that was used in the Ebola virus vaccine that I mentioned earlier that was produced so rapidly. And so what is an adenovirus vector? Adenoviruses are another type of virus, like the coronavirus, but they normally cause common colds. So the Ad26, which is a sort of flavor of adenovirus, has been disabled. So it can't replicate and therefore can't cause disease if introduced into a human through a vaccine. And so a vector is a circular piece of DNA, which is the genetic material of the virus that has been taken from the adenovirus. And this is used to transport pieces of the pathogen in order to stimulate an immune response. And so if we look at the diagram shown on this slide, you can see an orange strand labeled genetic material. And that represents the SARS-CoV-2 genome. And so what scientists do is they take a little bit of that SARS-CoV-2 genome called a gene. And in this case, the gene codes for S protein, which is unique to the SARS-CoV-2 and thus allows your immune system to produce antibodies to SARS-CoV-2. And so that gene for S protein is then added to the adenovirus genome, and that combination creates a stable plasmid. And so we can't just inject a gene for S protein into us because that would be too unstable. So we have to add it to that vector so that the whole product can stably be introduced to us through vaccination.
Interesting. Now, I heard that phase 1 clinical trials for this vaccine is not supposed to actually start until September. Why is that?
Yeah, so a lot of the other vaccines that Sam's mentioned today have already started phase 1 clinical trials, but this vaccine can't begin phase 1 because it takes time to create the adenovirus vector. And so that sort of delays the start date of phase 1 clinical trials. But once phase 1 is able to begin, it should only last about 6 weeks. So this is much shorter than the typical amount of time that phase 1 lasts for vaccines.
Gotcha. So if this does get approved by the FDA, how many vaccines are going to be able to be made?
Yeah, that's a great question. So as of now, the company which is working on this vaccine has one 200-liter vessel. And the company said they can make 300 million vaccines per one vessel. And so that would be enough vaccines to roughly vaccinate everyone in the United States. However, the ultimate goal is to be able to vaccinate everyone in the world. And so it's really important that this company and everyone is able to work with other global companies and use everyone's research so that if a vaccine is approved, it can be distributed globally and not just to one country.
Excellent. Well, it'll be interesting to see how this kind of develops over the coming weeks and months. Now, basically there have been two other coronavirus epidemics in recent history that most people, if they weren't familiar with them before, they are now. So the previous two being SARS and MERS. Um, was a vaccine actually ever made against either of those viruses? I haven't heard of the MERS vaccine or, a previous SARS vaccine. So what are we doing in that process, and how does that compare to what maybe with the challenges we have coming up in the future?
Yeah, so unfortunately, there were no vaccines created for SARS-CoV-1 and MERS-CoV, and there were a couple of reasons as to why that was the case. The first one was that there wasn't much evidence that these vaccines really worked. These vaccines created an antibody response, but to clear the coronavirus, another type of response called a T cell response may have also been necessary, and these vaccines didn't really activate that response. Another issue with past coronavirus vaccines were there were some safety issues. Some lab workers were potentially exposed to live virus, and they were worried about incomplete inactivation of the virus. They thought that this might lead to outbreaks. And then, quite frankly, the last reason that a past coronavirus vaccine wasn't created was that there was a lack of interest in funding. Researchers didn't really think it was a pertinent issue. The first SARS-CoV-1 outbreak just sort of died off on its own, and we only see a couple of cases of MERS per year. And so the scientists didn't really think the virus would reemerge, or they weren't sure that it would. And quite frankly, it takes a lot of money to produce a vaccine. Sam mentioned earlier, it's about $1 billion. And so if, a certain disease isn't deemed as a pertinent issue, researchers aren't going to be willing to invest that money to create the vaccine.
Wow. Yeah, it is. That is quite a figure. $1 billion is nothing to laugh at.
Yeah.
So with all of this information taken into account, if you were to sum up everything that we've talked about today, will a vaccine be available to solve this current SARS-CoV-2 pandemic?
Yeah, unfortunately, that probably won't be the case. Like we mentioned earlier, the typical vaccine development process takes about 10 years. This will be expedited, to a great degree. But still, even with that being the case, we're not going to have a vaccine ready for another 12 to 18 months. And this pandemic is projected to end before then. And so taking that into account, some people might question why we're even still doing research for a vaccine. And the answer for that is that we're creating a vaccine in case the coronavirus somehow mutates and comes back, or if the coronavirus becomes seasonal. We want to be prepared in case there is another outbreak. We don't want to shut down the world every time there's a coronavirus outbreak. And so that's why it's really important that we're still using our resources, coming together and finding a vaccine.
Excellent. No, I appreciate you bringing that whole picture kind of together.
Yeah. Absolutely.
Well, to wrap things up today, you've all heard from our team, and I really do appreciate all three of you for coming on the show. You've spent, many, many, many hours, putting this process together, and I appreciate your effort, your time, and educating your friends and family. Thank you again to all three of you for coming on the program, and best of luck as PA school starts here in a few weeks. I can't wait in a few years to see all of you as practicing providers. Thank you so much.